All right, so I'm doing pages 54 through 57. So we're going to start picking up from where Neil left off at the ATL skills. This section discusses skills that are important for reading this book and specifically analyzing research studies. And it suggests the RCE method. This method essentially means to focus on a study's aim, methods, procedure, results, and conclusions, as well as evaluating the strengths and weaknesses of the study. By doing this throughout the book, you can often fill in gaps where information might not be fully presented or given to you, and that helps you to further understand the research studies that this book actually talks about. The next section is conclusions. This picks back up on the localization and sort of just discusses localization and some more case studies that support localization. It mentions the Sperry and Gazzaniga split brain studies, as well as other studies, and then it goes into depth about the limits of localization. The first being lateralization, otherwise known as localized functions that are localized weakly. Localization is shared between several areas under lateralization, localization is shared between several areas, but is dominated by certain areas of the brain. Another limitation for localization are widely distributed functions, where the function is distributed throughout the entire brain instead of being localized in one place. There are also cases where some components are localized while some are distributed. This can be seen with language, where certain components are localized in the Broca's area or the Wernicke's area, while other components are more broadly distributed. And then the last point it mentions is neuroplasticity, which essentially in relation to localization is when a function sort of moves from one part of the brain to another. This is most often caused by injury, where one part of the brain is damaged. One part of the brain that does, say, language or does the Broca's area, for example, is damaged. Another part of the brain will start to attempt to pick up that function that part of the brain served. Much like localization, neuroplasticity is also relative and has limitations, and this gets discussed in the next section. A last point from this section is that generally localization is found to be more strict with relation to functions that are sensory or motor based, and more lateralized or distributed in higher cognitive functions like memory, thinking, or learning. So the next set of pages are going to talk about neuroplasticity. Neuroplasticity, as I sort of mentioned before, is the ability for the brain to change by breaking and making synaptic connections. This is caused by both genetic and environmental factors. Neuroplasticity scales from synaptic to cortical remapping. Synaptic plasticity is entirely dependent on the activity of neurons. This section also discusses neuroplasticity as a learning mechanism. Neuroplasticity does occur on a daily basis as we learn new things. This was proven through Dragensky's research study and McGuire's research study. Practical applications of neuroplasticity is also discussed in this section. This can be seen in sense substitution or echolocation, like human echolocation, or brain-machine interfaces. Lastly, this section links to localization, cognitive processing, and brain development. All right, lastly, page 57. This starts by discussing some definitions, including going back over the definition of neuroplasticity that I mentioned earlier. It also defines synaptic plasticity, which is the ability for neurons to form new connections and break old ones, as well as cortical remapping, which is when brain area A assumes the function of brain area Y, most often due to injuries. And then this page talks about the Merzenek et al. 1984 research study and remapping of the sensory cortex. So the Merzenek et al. Research study was a study that was conducted on 8 adult owl monkeys. This was done in 3 steps. The monkeys' hands were measured. They were wired to electrodes that would measure sensory inputs in their digits, in each of their digits. For the second step, they amputated the middle finger, and then for the last step, they waited 62 days and then measured again to find remapping in the monkey's hands. What this study found is that each digit has an individual location in the cortex with adjacent fingers or digits having adjacent locations. The areas that corresponded with the second and fourth fingers, the ones adjacent to the amputated third, expanded into the now unused third area in the brain, or in the cortex. This allowed them to come to the conclusion that cortical remapping had taken place in the owl monkeys over a period of 62 days.